Okay, so let's get started. Hello, everyone. Welcome our today's Amy Zoom seminar. Again, first of all, I want to thank everyone being with us during the weekend on our seminar series, and of course, special thanks to our today's speaker, Dr. Omar Yilmaz from MIT. I mean, thanks Omar for taking the time during the weekend to give us his lecture, share the science with us today. Um, so before I start the seminar, I just want to give a very brief introduction about Oma. So Oma finished his undergraduate study in biochemistry and physics in University of Michigan, and then continue with MD training also in University of Michigan Medical School. And after that, he pursued his PhD training in cellular and molecular biology, also University of Michigan Medical School. So, so from 2011, Omar actually moved to Boston to start his PhD postdoc training in Whitehead Institute for Biomedical Research. And then 2014, Omar started his independent research program as assistant professor in biology in MIT. Um, so from 2022, he's promoted as an associate professor of biology with tenure. And then last year, he's also been appointed as director of the MIT Stem Cell Initiative. So OMAS is a physician scientist with a research program dedicated to understanding how diet profoundly influence intestinal health and disease. His lab mainly interested in the biology of intestinal stem cell, particularly how they integrate dietary cues to balance self-renewal and differentiation, ultimately shaping intestinal composition and function. A major focus of his research is the interaction between the intestinal stem cell and their surrounding niche, particular penicell, and how this relationship is modulated by nutritional signals. Uh, his lab aimed to uncover the molecular machinery by which diverse dietary regions influence stem cell activity, niche function, and then regenerative capacity, with implication for the formation and progression of intestinal malignancies. I just actually mentioned that I'm actually currently traveling, but I told him Omar that I definitely doesn't want to miss his seminar because I'm actually really a big fan of his work. He actually did really fascinating work on the intestinal stem cell. And then with all these achievements, I just list a few of awards and the honors he received, which including the AAS Martin and Rose Watchell Cancer Research Prize, Eisen and Chen Career Development Professor in MIT, Crohn and Colitis Foundations as a Senior Research Award from 2023, and Kenneth and Reining Foundation for Innovative Award, also 2023. So I think today Omar is so going to talk about some past work and then recent work from his lab regarding the dietary components, how they control and influence the intestinal stem cells. So for that, thank you so much, Omar, being with us today, and then we are very much looking forward to your talk.
Well, Tron, thank you so much for the kind introduction, and it's a pleasure to be here today. Actually, I love giving talks on Sunday because it's usually nice and calm, so it's a great time of the week to really think about science. So, as Tron mentioned, I've my lab's had a long-standing interest, actually dating back to my time as a postdoc in David Sabatini's lab, to really understand how diet impacts intestinal stem cells and physiology and disease. Let me see here. So before I get started, I just like to acknowledge the trainees' work that I'm going to be talking about today. So today is going to be a mix of both recently published work, but there's also going to be a lot of unpublished work. The unpublished work is going to be work from Swagata Goswami, Ximing Zhang, and then at the end, I'm going to talk about some really exciting work by Feng Tao Qi, looking at the effects of amino acids on stem cell self-renewal. And some of the published work that I'm going to talk about will be recent work that we've done on fasting refeeding by Shinya Imada and Saleh Kawalid. So when I started my lab back in 2014, I was really interested in the question, how does organismal diet impact stem cell function and physiology and disease? And in the lab, we interrogate this by looking at extreme types of diet. At one end of the spectrum, I've always had a long-standing interest in understanding the effects of low-calorie interventions such as caloric restriction and fasting, in part because these low-calorie interventions in multiple species have been shown to have anti-aging and in some mammalian studies, anti-cancer effects. And more recently, after starting my lab, we became much more interested in obesity and pro-obesity diets, given that there's been long-standing epidemiologic studies implicating obesity and pro-obesity diets to diseases of aging, such as cancer. So diet is known to have a strong effect on regeneration, aging, and diseases such as cancer, but we still don't know a lot of the cellular mechanisms and pathways that regulate this in given tissues, such as the intestine. So I'm going to start off by talking about our work on obesity. And as I mentioned, the reason why we're interested in obesity is several-fold, but there's a lot of epidemiologic studies linking obesity to cancer incidence and mortality in a broad range of tissues. One of the more prominent of these tissues is the colorectum, which is what we study in my lab. And when I started my lab, we asked a very simple question. How does obesity impact colon cancer incidence and progression? So, just briefly, in the United States, colon cancer is the second leading cancer cause of death for both men and women. Every year, there's about 150,000 new cases diagnosed. And unfortunately, about 50,000 patients die from complications of obesity. I mean, from colon cancer, which predominantly is driven by metastatic disease to the liver and peritoneal spread to the abdominal cavity. And an interesting observation over the past 20 years is that the incidence of colon cancer in young patients, namely patients younger than the age of 50, has increased dramatically, really raising the possibility that there may be a role for diet and obesity in this process. And we actually are part of a CR UK team that includes Yasmin Balcade, which is led by Andrew Chen and Yin Zhao at MGH and WashU, respectively, to really delve deeper into, at least our lab, to delve deeper into some of the mechanisms by which obesity and other exposures might be contributing to this process. As I've mentioned to you, our model system for addressing these types of questions is the mammalian intestine, shown here in schematic. The intestine is really the simplest tissue type that one can study in the body. By definition, it's a simple epithelium, meaning that it's a single layer of epithelial cells. In the small intestine and colon, the simple epithelium is folded upon itself to form these cryptomains. These cryptomains are the workhorse of the epithelium, and at the bottom of these crypts are the stem cells, shown here in green, which are LGR5 positive, originally demonstrated by Hans Clevers back in 2007. These LGR5 positive stem cells give rise to progenitor cells, but there's tremendous plasticity in the intestinal lining where, during damage, for example, cells higher up in the crypt, such as these upper crypt cells, or even cells that are intermediate between the upper crypt and the crypt base, can get reprogrammed and repopulate the stem cell population in supraphysiologic conditions. So the reason why we're interested in studying these LGR5 positive stem cells is that in model organisms, and there's evidence that this is likely to be true in humans, that these long-lived stem cells accumulate oncogenic mutations throughout the life of an organism and give rise to stem cell initiated cancers. So we really want to understand how these different exposures change the property of these stem cells and render their vulnerability for accumulating and undergoing oncogenic transformation. So now that I've given you a little bit of a background, what is the role of diet and obesity in the early steps of intestinal tumor formation? So when I started my lab in 2014, we've really spent this has been a continuous project in my lab, as you're going to see. So with respect to normal diet, as I mentioned to you, long-lived stem cells repopulate the epithelium, and over the life of an organism, accumulate oncogenic mutations to engender stem cell-initiated cancers. So what happens in obesity? So there's many ways to model obesity. A common way in the field is to place mice on a long-term high-fat diet where these mice derive about 50 to 60% of their energy from animal fat, 20% from carbohydrates, and 20% from protein. Within a time frame of a couple months, these mice become obese. So what happens here in a long-term high-fat diet after being on this diet for close to 1 year? What we find are two important changes. Number one, in a long-term high-fat diet obesity model, there's an expansion in the number of LGR5 positive stem cells here shown in green. Number two, in this long-term high-fat diet, progenitor cells higher up in the intestinal crypt, which normally lack the ability to self-renew, start to behave like stem cells in functional assays. And the upshot of these changes of having more LGR5 positive stem cells and progenitor cells that have acquired functional attributes of stem cells is that with respect to cancer, one has increased the pool of target cells that can undergo oncogenic transformation, such that in the presence of oncogenic mutations in the high-fat diet, one can find both stem cell-initiated cancers as well as now progenitor-initiated cancers. And over the years, we've spent a lot of time trying to elucidate the mechanism undergirding this observation. And I'm just going to give a one-slide summary, since this work has been published. But we propose that there's at least two non-overlapping mechanisms that contribute to this phenotype. Number one is intrinsic to the stem and progenitor cells, where we propose that lipids associated with the diet or associated with obesity can engage and activate this family of transcription factors called PPARs, which can drive fatty acid oxidation. We find that this PPAR fatty acid oxidation program is important for expanding stem cell numbers and for endowing stemness to progenitor cells. And independent of this pathway, what we found, consistent with what others have shown, is that in a long-term high-fat diet, there's a reduction in the microbiome diversity. This reduced microbiome diversity associates with reduced MHC class II expression on premalignant stem and progenitor cells, enabling these early premalignant cells to evade CD4 positive mediated T cell detection and elimination. So we've been very interested in studying the beginnings of how obesity can regulate the early steps of tumor formation. In addition to the high-fat diet, we're now looking at ketogenic diets as well as more carbohydrate-based obesity models. And to really further our understanding beyond the beginnings of tumors, we wanted to ask the question, how does a high-fat diet-induced obesity model impact colon cancer progression and metastasis? Clearly, obesity enhances the early steps, but what about the latter steps, which accounts for the vast majority of morbidity and mortality in patients? And over the years, this has been a very hot topic. There's been a lot of interest in the cancer field, particularly in the colon cancer field, for identifying the cells and primary tumors that initiate metastasis in the liver. So original work by Fred Savage and colleagues at Genentech proposed that LGR5 positive tumor cells from the primary tumor extravasated into the vasculature and were the earliest cells that seeded metastases in the liver. Over the years, our understanding of this phenomenon has become more nuanced, where work by Jakob van Rijn and others have actually shown that the cells that likely initially set up shop in the liver are LGR5 negative cells, which account for the vast majority of tumor cells. But upon setting up shop in the liver, these cells immediately revert to an LGR5 positive state, which in some ways reconciles their finding with the earlier work by Fred Savage and colleagues. and more recently work by Kruna Ganesh and Edu Bhatia at MSK and Barcelona, respectively, have shown that EMP1 positive cells, which for the most part are LGR5 negative, are the subset of LGR5 negative cells that home into the liver and initiate metastatic seeding and outgrowth. So where do we fit in? So clearly, we now have some insight into what are the cell types that are leaving the primary tumor to initiate metastasis. Our question was, can one identify metabolic or signaling pathways that drive the acquisition of this population in the primary tumor? And we really wanted to test this out in our obesity model with the hope that by placing these mice on a supraphysiologic obesity model, we might really accentuate the discovery of important pro-metastatic metabolic pathways that can lead to the acquisition of these metastasis-initiating cell state phenotypes. So here we place mice on a control diet, and we use a defined control diet. This isn't a chow diet. And we use our defined high-fat diet. We age recipient mice for 8 weeks. We then transplant these mice with colon organoids that harbor mutations in the KRAS oncogene, loss of function mutations in APC and P53. This APC KRAS P53, which I'm going to use the acronym AKP for, is a very important genotype because it's found in up to 45 to 50% of all human colorectal cancers. We then transplant this using mouse colonoscopy, which was a technique really optimized and in some regards pioneered by a previous trainee in my lab, Jayton Roper, who now runs his own lab at Duke Medical School. And then we, after a few weeks post-transplantation, these mice develop primary tumors. So the question is, what about liver metastases? Does this obesity diet impact that process? And surprisingly and not surprisingly, the answer to that question is yes. Recipient mice on a high-fat diet die much sooner than their control diet counterpart, and this nicely associates with a significant increase in numbers of metastatic lesions in the liver. And an important piece of data is if we did these transplants in conventional mice that had a normal adaptive immune system, if one repeats these experiments in mice, in RAG2 mice, for example, that lack mature B and T cells, one comes to the same conclusion, indicating that high-fat diet-induced obesity promotes colon cancer metastasis independent of the adaptive immune system. And I think this is important because here we use a physiologic genotype, a physiologic transplantation model to come to these conclusions, because in the literature, there's some papers that suggest that perhaps the adaptive immune system in this particular high-fat diet model contributes to the phenotype. We don't find that with respect to metastases. So we wanted to gain some insight into what could be happening here. So we performed bulk and single-cell RNA sequencing on the high-fat diet and control diet primary tumors. We identified, of course, many interesting gene signatures, but one gene signature really caught our attention, which is this revival stem cell signature originally proposed by Jeff Rana and colleagues at the University of Toronto, where they showed in the normal intestine, though the normal intestine is maintained under homeostatic conditions by LGR5 positive stem cells, since these LGR5 positive stem cells are quite proliferative, upon chemotherapeutic or radiation-induced damage, this proliferated population gets severely compromised. And what Jeff Rana and colleague proposed is that more differentiated cells higher up in the intestinal crypt in a YAP1 dependent manner can activate this YAP1 program, which some people call a revival stem cell signature, others call it a fetal program, others call it a reparative program. For the purposes of my talk today, I use revival stem cell signature, but I think from here on out, we're just going to call this a YAP1 reparative gene signature. But the upshot of all of these names is that a cell higher up in the intestinal crypt in a YAP-dependent manner in severe injury can repopulate the stem cell zone at the bottom of the intestinal crypt. And this holds true not only in the normal intestine, but work by Owen Sampson and others have proposed that this same revival stem cell signature or YAP program is present in colon cancer. And just to remind you, really briefly, that YAP is a transcriptional coactivator that's sequestered in the cytoplasm upon environmental stimuli or even intracellular stimuli. YAP can then translocate into the nucleus and bind to the T transcription factor and drive in the intestine this regenerative fetal revival stem cell program. And again, in colon cancer, there seems to be a lot of stem cell plasticity between the CRIP-based cells, which are the WNT program or the LGR5 cells, and this more YAP1 dependent population, which has been proposed to play an important role in resistance and metastasis. So what is the impact of the high-fat diet on this YAP revival stem cell program? So here we're looking at our single cell of the primary tumor, and for the purposes of this story, we've only focused on the primary tumor, not on the Mets themselves. We're hoping to study the effects of high-fat diet on the Mets in future studies. So in our single cell studies, what we find is that this YAP1 signature revival program is located at the base of what looks like the continent of Africa. We use one marker, BAS1, as a proxy for this revival stem cell signature, and one can appreciate here in terms of enrichment. In the high-fat diet, there really is enrichment of this revival population. And this isn't some artifact of single cell sequencing because if one performs in situ hybridization for BAS1, one again finds that within the high-fat diet tumor, there's strong induction of BAS1 compared to the control diet counterparts. I want to mention that I'm not saying that there is no BAS1 positivity in the control diet. There clearly is, but the relative ratio of BAS1 positive cells goes up notably in the high-fat diet. And similarly, one can make associations in our single cell data set with YAP1. So again, here BAS1 associates with canonical YAP1 target genes such as CCN2. And I want to point out that this population is distinct from the LGR5 positive tumor cells. Again, in accord with what's been proposed by Owen Sampson and others, that there seems to be mutually exclusive populations between LGR5 positive cancer cells and the cancer cells that are high for the YAP1 program. And again, the YAP signature is elevated, and by immunohistochemistry, one appreciates more nuclear active YAP1 in the high-fat diet compared to the control. So do these YAP revival stem cell programs drive metastatic growth and seeding? To address this question, we took advantage of back recombinering, where we took the back for BAS1, we placed the GFP DTR cassette into the BAS1 promoter. So BAS1 positive cells should be both GFP positive and should express the diphtheria toxin receptor, rendering them susceptible to diphtheria toxin injections for depletion purposes. And one important experiment that we did here is we transplanted our AKP organoids that harbored our BAS1 cassette. We let tumors engraft for about 4 weeks, and then we inject these mice with diphtheria toxin to understand what role BAS1 positive cells play in metastatic seeding. And surprisingly, what we find here is that when we deplete the BAS1 positive cells with diphtheria toxin, we essentially ablate, if not severely dampen, metastatic seeding in the liver. And again, we've done these experiments in control diet and in high-fat diet mice and come to the same conclusion. And moreover, if one looks at the size of the primary tumor, upon BAS1 depletion, surprisingly, the primary tumors for the most part are the same size, which is similar to work that's been done with LGR5. If you deplete LGR5 positive cells in primary tumors, the primary tumor size doesn't change. So this really raised the question of, can we identify metabolic drivers in the primary tumor? We performed the following serial transplantation experiment where we took where we generated organoids from tumors from control diet and high-fat diet mice. We call these primary organoids. We then passage these in control diet mice and generate secondary organoids. And I should point out here that these primary and secondary organoids have been now many weeks, if not many months, outside of the high-fat diet milieu. So how do they behave? So interestingly, we performed a lot of omics. We performed lipidomics, RNA-seq, attack sequencing, and I always tell trainees it's great to do omics, but don't forget economics, um, because this ends up costing a lot of money. In this particular case, we were lucky because we were able to identify some very interesting biology and just functionally to show you that despite the fact that these primary and secondary organoids have been outside of the high-fat diet milieu, these high-fat diet-derived organoids are still locked in a pro-metastatic state. So this pro-metastatic state, upon exposure to a high-fat diet, seems to be durable, which is very interesting. So what is the is there a durable metabolic phenotype that's driving this effect? So in collaboration with Alparslan Taşdoğan in Germany at Essex University, we performed lipidomics, and surprisingly, we find that in terms of lipidomics, that the high-fat diet-derived organoids are clustering quite differently from their control diet counterparts. And if one looks at volcano plots, one finds that it's really one particular species of lipids that seems to be really driving this difference, which are long-chain ceramides. So long-chain ceramides seem to be elevated in the high-fat diet-derived organoids compared to their control diet counterparts. And just to remind you about ceramides, they're a very active class of lipids. They have important signaling properties, and they essentially are derived from serine and palmitoyl-CoA through a series of enzymatic reactions to generate ceramides. And ceramides themselves can feed into the generation of other lipid species, such as sphingomyelins, sphingosine, and glucosyl ceramide. We really focused on ceramides for many reasons. Number one, downstream lipids of ceramides didn't come up in our lipidomics. Number two, when we looked at our gene expression data, it was really the ceramide producing enzymes rather than ceramide product enzymes that really came up in our high-fat diet signature. So again, DEGS1, which is the enzyme preceding ceramide generation, also associated with our BAS1 revival population, as did many of the other upstream enzymes. And again, DEGS1 itself, by in situ hybridization, seems to be highly enriched within the high-fat diet tumors compared to the control diet. So is there a role for ceramides in metastasis? So we went ahead and deleted DEGS1 in our AKP organoids, and as expected, when we knock out DEGS1, this leads to a reduction in both our number one, it leads to a reduction in ceramides, which I'm not showing you, which also corresponds to a reduction in our YAP and revival stem cell signature. And most importantly, functionally, what we find here is that when we ablate DEGS1 and transplant these AKP organoids into control diet and high-fat diet mice, one finds that loss of ceramide production essentially nullifies metastatic ability in these AKP organoids. Again, the primary tumors are essentially the same size, but loss of DEGS1 now severely hampers the ability of these cells to form metastases in the liver. And I'm not showing you ceramides are critical for YAP activation. We think that these findings are relevant to humans. Fortunately, now there's a lot of omics data, especially single-cell RNA sequencing from patient samples. So when we look at the data set from Nir Hacohen's group at MGH, and we were able to get the metadata associated with BMI, what we find here is that both this revival stem cell signature and YAP target gene are elevated in primary tumors from these patients. And again, one finds elevation of genes involved in ceramides, such as DEGS1, DEGS2, SIRS S2, SIRS S6. And lastly, when we look at Karuna Ganesh's data set from MSK, we again find that there's nice overlap between our revival stem cell program, YAP1 target genes, and DEGS1, as well as other ceramide producing enzymes. So to summarize this part, we are proposing that tumor-derived ceramides tumor intrinsic ceramides are important because they activate a YAP revival stem cell program, which enables metastasis. And I think we got really fortunate here because by placing mice on a high-fat diet, we were really able to drive the detection of this signature for this program that we think is not only operative in our obesity model, but also operative in the control diet state. So we're really interested in going deep with how ceramides, for example, are activating YAP and really getting a better understanding of whether this program is also operative within metastatic lesions themselves. So I'd like to shift gears a little bit and talk about work that we've recently done with fasting refeeding, which is the opposite end of the spectrum. As many of you know, low-calorie interventions have shown to promote lifespan, health span, and/or health span in a broad range of organisms, from worms to mice, and even in some primate studies. And a few years ago, we asked a very simple question. How does 24-hour fasting or deprivation of food impact stem cell function? So here we took advantage of the organoid assay where we could sort stem cells and cells. Cells are these chubby cells next to the stem cells. They're part of the epithelial niche for stem cells in the small intestine and proximal colon. And we can assay uh, the ability of stem cells to give rise to these mini organoids or mini intestines in culture. And what you can see here is after a 24-hour fast, stem cells are nearly twofold more functional than their ad libitum counterparts. And this is independent of co-culture with the niche. So fasting is activating some pro-regenerative program in stem cells. So what is that program? Interestingly, we propose that in fasting, what's happening here is that, given that these mice are deprived of carbohydrates, mammals will undergo lipolysis. They'll liberate fatty acids into the circulation. And we propose that these fatty acids, by activating these PPARs, which are lipid sensors, activates fatty acid oxidation, which is important for which is an important mechanism for these stem and progenitor cells to derive energy for persisting during times of nutritional stress. So, we found a few years ago that fasting was important. It activates and preserves stem cell activity in a fatty acid oxidation dependent manner. We are able to mimic this synthetically by treating mice with PPAR activators. PPAR activation seemed to mimic some of these pro-growth effects. But we were always left with an open question after these findings. What's really driving stem cell mediated regeneration? Is it fasting or post-fast refeeding? Because if you remember, our assay for looking at this was the organoid assay, where we took fasted stem cells and we placed them into nutrient replete conditions. So it was always a formal possibility that fasting, it wasn't really fasting itself, but it was the post-fast refed state that was really driving this regeneration. So, so the way we think about this is that fasting is important. In fasting, stem cells activate fatty acid oxidation, and this is important for persistence and survival. And it could be formally possible that post-fast refeeding is what's really inducing stem cell mediated regeneration. So we addressed this question doing very careful in vivo mouse experiments where we generated cohorts of mice that were either ad libitum fed, fasted for 24 hours, fasted and refed for 24-hour intervals, and then fasted for 24 hours and refed for a 3-day period. And at the end of this analysis, we looked at many different outputs, but the one output that was most interesting was the BRDU proliferation assay, where we found, consistent with our earlier work, that a 24-hour fast notably decreases proliferation within the stem and progenitor cells. But interestingly, one day after refeeding, proliferation now goes up significantly. Not only is it greater than the fasted state, but it's also greater than the ad libitum state. But by 3 days of refeeding, now proliferation goes back to the ad libitum baseline. So this was really interesting to find. So what about stem cell quality? So stem cell numbers don't change after a 1-day fast refeed, but what about the quality of these stem cells? So here we took advantage of an LGR5 TD tomato reporter, where when one injects these mice with tamoxifen, one indelibly labels the stem cells with TD tomato, enabling one to track stem cell output. And then here we again looked at our different cohorts of mice, and we damaged the intestine with radiation to really select for a regenerative phenotype. And what we found here, surprisingly, was when we damaged the intestine 1 day after refeeding, we see the greatest number of TD tomato output compared to fasting and the ad libitum state. And again, this phenotype disappeared 3 days after refeeding, which really leaves us with the following paradigm where the post-fast refed state, the stem cells are in a supercharged state to repopulate the intestinal lining after injury. So what is the underlying mechanism here? We immediately turned our attention to the mTOR pathway, given that mTOR is really a master regulator of nutrient sensing. There's great pharmacologic and genetic tools to really distill out mechanism. And when we sort out stem and progenitor cells and look at mTOR activity, we again find that 24 hours after refeeding, mTOR activity is really high. So both in stem cell and progenitor cells, looking at multiple output of mTOR activity, we see very nice and strong mTOR activation. So what happens if we block mTOR activity during the refed state? We've done this by looking at Raptor knockout mice or treating mice with rapamycin. The Raptor mice data, which I don't have in here, is even more impressive. But if we treat mice with one injection of rapamycin during the refed state, we can now attenuate this refeeding response that we observe after injury in the intestine. So clearly mTOR signaling here is very important at driving this regenerative state. So we believe that fasting, fatty acid oxidation are important for survival, and that mTOR activity, and in data that I'm not showing you, we think insulin is also important, which feeds into mTOR activity. But the question is, what is it downstream of mTOR that's really reinforcing this refeeding phenotype? So to gain insight into this, we performed single cell sequencing, and we added a new cohort here where we treated our refed mice with rapamycins before initiating refeeding and during the refed state. We performed single cell analysis on the stem and progenitor cells. We identified different progenitor cell clusters, including 3 stem cell clusters, which was previously shown by Aviv Regev and Ramnik Xavier, and based on LGR5 positive expression as well as other stem cell markers. Cluster 5 is the most primitive, followed by 2, followed by 10. So when we perform differential gene expression on these different stem cell clusters between, refeeding ad libitum or refeeding with rapamycin, we saw a lot of interesting genes change, but there was one metabolic enzyme that always seemed to be up in the refed state in an mTOR-dependent manner, and that is the enzyme ornithine amino transferase, which I'm going to get into. But you can just see here again by in situ hybridization, OAT is expressed in the intestinal crypt. It doesn't change with fasting, but in refeeding, its levels go up in an mTOR-dependent manner. So if you treat with rapamycin, it goes back to baseline levels, which is in accord with what we saw in our single cell RNA sequencing data. So what is OAT? OAT is a metabolic enzyme that interchanges proline glutamate with ornithine and vice versa. And it's interestingly most abundant in the intestine. And in data that I'm not showing you, when we perform metabolic profiling, ornithine itself goes up in the refed intestinal stem and progenitor cells. We were really interested in this because I don't want to get into all of the metabolic details since I know this audience is more immune focused, but ornithine really has 2 fates within a cell. One fate for ornithine is that it can feed into the urea cycle. The other important fate for ornithine is that it can feed into the polyamine metabolite pathway. And when we looked at gene expression data, we saw induction of many of the enzymes, including rate-limiting enzymes for polyamines, and not for the urea cycle. So we initially focused on the polyamine pathway where ornithine, through a series of enzymatic reactions, can get converted into putrescine, spermidine, and spermine. And again, we perform metabolite profiling. We find that refed stem and progenitor cells show elevation of these polyamines, such as spermidine. And again, if we look at rate-limiting enzymes by qPCR or by single-cell sequencing, ODC1 and SRM and AMD1, all rate-limiting enzymes are elevated in the refed state, particularly in the acute refed state. And there's an interesting property about polyamines, in particular spermidine. It turns out that spermidine plays an important role in the regulation of aspects of protein synthesis, where spermidine, through enzymatic reactions, can post-translationally modify elongation factor 5A, which enables the translation of mRNAs in the immune system. Work by Erica Pierce has shown that this associates with genes involved in stress and inflammation. And again, an independent way to validate our data is that in the refed state, one finds more hypocinated elongation factor 5A, again, in parallel to what one would expect with having an increase, in agreement with having more polyamines present in the stem and progenitor cells. So what happens to protein translation? Excuse me, mRNA translation. So here we perform pyrimycin assays where we isolate crypts from the different conditions. We treat with pyrimycin and look at protein production. And again, here you can appreciate that that RNA translation goes up in the refed state compared to fasting refeeding, and it goes up in an mTOR and polyamine dependent manner. So if you treat with rapamycin, if you knock out Raptor, or if you treat mice with DFMO, which inhibits ODC1, which is the first committed rate limiting step of polyamine production, we can now dampen and prevent this increase in mRNA translation. And again, we've now made ODC1 conditional mice where we can ablate ODC1 just in the epithelium and we come to the same conclusions. So really putting it all together, we propose that fasting refeeding represent two distinct metabolic states, where in fasting fatty acid oxidation is important for stem cell survival, and in refeeding this mTOR polyamine protein synthesis axis is really important for regeneration. And in hindsight, this makes a lot of sense because fasting is a catabolic process, whereas refeeding is an anabolic process. So in the refed state, the stem cells are trying to repopulate and turn over the intestinal epithelium. So what about tumor formation with respect to fasting refeeding? This is an interesting question. I think the dogma in the field is that low-calorie interventions are almost inevitably good in terms of cancer prevention. But actually, if you take a step back and you comb through the literature, it's not that straightforward. In fact, studies that use the same mouse models come to different conclusions with respect to caloric restriction and in the APC-min model. Even outside of the intestine, when one looks at lymphoma incidence, different studies come to different conclusion in genetically identical mice. And we think that some of the confusion here may stem from the fact that it remains unclear how the timing of mutagenesis with respect to fasting refeeding cycles influences tumor formation. So again, we took advantage of our defined mouse models where we can use APC LOX LGR5 mice. When you inject these mice with tamoxifen, one deletes both copies of APC, which is a tumor suppressor gene that's lost early on in colon cancer in the intestinal stem cells and gives rise to adenomas. When we delete APC in our different cohorts and then analyze the intestines, what we find here, this at the time was very surprising, but it makes a lot of sense. Both in the small intestine and in the colon, we find that loss of APC 24 hours after refeeding leads to about a twofold increase in tumor formation. And we actually found here that deleting in the fasted state really had no anti-tumor effect. And again, we can do this with respect to polyamines. In the same models, if we now treat with DFMO, which inhibits polyamine production, or if we do this with genetic loss of ODC1 or dampen protein translation with cyclohexamide, we come to the same conclusions where, for example, if we block the increase in polyamines, we now block the increase in tumor formation, both in the small intestine and colon. So I just want to summarize and say that again, in the fasted state, having less nutrients turns off mTOR, and mTOR, the major regulator of protein translation. So protein production goes down. These cells switch to fat metabolism for survival. And in the post-fast refed state, the abundance of nutrients can activate mTOR and this polyamine protein production pathway, which leads to anabolic recovery in the intestine. But if you're unlucky and you get exposed to a mutagen or to a carcinogen in the refed state, at least in our mouse models, this may prime you for early tumor formation. And in work that I'm not showing you, Feng Tao Chi, a very talented postdoc in the lab, he's now identified amino acids and transporters for a particular amino acid that feeds into polyamines. And if he knocks out that mitochondrial transporter, and he's now working on diets that are lacking in that amino acid, he's able to blunt some of the tumorigenic effects that we see associated with refeeding. And now that I mentioned Feng Tao, I have a short vignette of what I think is a really interesting story. And this is really going to be the next phase of my laboratory. Really the next 10 years of my lab is going to be devoted to identifying how micronutrients regulate stem cell activity. Up until now, we've taken a whole dietary approach, obesity, low-calorie interventions, and we're still going to study those at high level because there's still a lot to learn there. But I'm really interested in identifying how micronutrients regulate stem cell fate. And a few years ago, when Xiaowei Cheng, who's now faculty at Columbia, was in the lab, she identified this metabolic enzyme, HMGCS2, which is the rate limiting step in ketone body formation. And she showed that HMGCS2 is a metabolic enzyme that corresponds to primitiveness in the intestine. Stem cells are high, progenitors are low, and differentiated cells even have lower expression. And if you place these mice onto diets that we know boost stem cell activity, such as fasting or high-fat diets, one finds that HMGCS2 levels go up. So we've been really interested in using HMGCS2 along with other markers as kind of a marker for stem cell activity. And the reason why this is important is it allowed us to ask the following question: how do amino acids regulate intestinal stemness? So Feng Tao Chi, he he uh, he gave IP injections of each of the proteinogenic amino acids to mice and screened for HMGCS2 activity as well as other markers that we're interested in. And you can see here there's a broad range of response with respect to each amino acid. Some amino acids really drive HMGCS2. Many have no effect, and some actually suppress HMGCS2 activity. So as proof of concept, we really focused on cysteine to get started with because cysteine really boosted HMGCS2 expression throughout the intestinal crypt and even in the proximal or lower parts of the villi. But we're now in the lab, we're now actively studying all of these amino acids. So to understand whether this increase in HMGCS2 with cysteine actually represents an increase in stem cell activity, we took advantage of the LGR5 TD tomato reporter. Remember, we inject these mice with tamoxifen and then damage the intestine with radiation. We can measure stem cell output by staining for TD tomato. And you can see here when we treat mice with a cysteine-rich diet and then damage the intestine, the cysteine-rich diet leads to about a twofold increase in stem cell output. And this raises the question, is the cysteine-rich diet acting on stem cells directly or non-autonomously, indirectly? And this was really exciting because when we started to examine the intestines from these mice, we noticed that there was a interesting increase in the number of intraepithelial CD8 positive T cells in the cysteine-rich diet. And this was interesting because it raised the possibility that perhaps CD8 positive T cells were mediating some of these effects. So Feng Tao repeated these experiments in RAG2 mice. And when he did, he found that the cysteine-rich diet lost its regenerative effects. So here we're staining for olofactomedin 4, which is a marker that's co-expressed in the LGR5 positive stem cells. And you can see here there's no increase in stem cell numbers after injury. So this really raised the possibility that likely adaptive immune cells, namely CD8 positive T cells, are in some manner responding to the cysteine-rich diet. So what about mucosal CD8 positive T cells? To get a more granular understanding of what's happening, Feng Tao sorted out the CD45 positive cells. And interestingly, what he found is that the proportion of CD8 alpha beta T cells was enriched in the cysteine-rich diet compared to the control diet counterparts. And then if he depleted the CD8 alpha beta T cells with antibodies and then placed these mice on a cysteine-rich diet, he again was able to nullify the effects of the cysteine-rich diet. So when he used control IgG, the cysteine-rich diet led to a big increase in stem cells after injury. And again, this was ablated when CD8 alpha beta T cells were depleted. So clearly, CD8 alpha beta T cells, a population that we don't normally think about as regulating mucosal or epithelial stem cell biology, seems to be playing an important role. So the mechanism here, so we performed unbiased cytokine arrays to really understand how the cysteine-rich diet was regulating the interleukin and chemokine, cytokine milieu of the intestine. And we observed some interesting changes, including in IL-22. And the reason why IL-22 caught our attention, as I'm sure many in the virtual audience will know, IL-22 has been shown by many groups to be a trophic factor for intestinal stem cells. So IL-22 through JAK-STAT signaling has been shown to really drive stem cell mediated repair in injury. So is IL-22 coming from our mucosal CD8 alpha beta T cells? Because historically, the main source of IL-22 has been suggested to be innate lymphoid cells. But what about in a cysteine-rich diet? And what about CD8 alpha beta T cells? So one of my favorite experiments is Feng Tao we obtained IL-22 knockout wild type mice. He isolated CD8 alpha beta T cells from the mesenteric lymph nodes and then adoptively transferred these to RAG2 deficient mice, which lack B and T cells. He then placed these mice on a cysteine-rich diet, damaged the intestines with radiation, and analyzed the intestines after 3 days post-injury. And as you can see here, when we have very nice engraftment of the intestinal mucosa with our CD8 alpha beta T cells. And when we look specifically at the stem cell compartment, compared to the control, mice transplanted with wild type CD8 alpha beta T cells show strong induction of stem cell markers, which is gone when we transplant these mice with CD8 alpha beta T cells that are deficient in their ability to make IL-22. So clearly, IL-22 is driving the regenerative effects of dietary cysteine and CD8 alpha beta T cells. So how? What is the mechanism by which cysteine-rich diet is engaging an IL-22 program in CD8 alpha beta T cells? So here we perform metabolic profiling on the small intestine and colon. And we observed many interesting changes, but one pathway that really caught our attention was this expansion or increase in the pool of CoA metabolites. And in data that I didn't show you, we knew CoA was likely going to be important because we weren't able to emulate the effects of a cysteine-rich diet with other known outputs of cysteine, such as taurine or glutathione. So that really led us to focus on CoA. And if we look at the structure of CoA, and I had forgotten this when we had started, when we had made this observation, but interestingly, there's a cysteine moiety at the end of the CoA. We asked a simple question: does CoA supplementation mimic the effects of a cysteine-rich diet? So the answer to this question is yes. So when we give CoA to mice, we see strong induction of HMGCS2 by Western blots as well as in our intestines by immunohistochemistry, consistent with the notion that CoA is boosting intestinal stem cells. Cysteine-rich diet, CoA supplementation increases IL-22 production as well as CD8 alpha beta intraepithelial T cells. And then most importantly, functionally, if we give mice CoA, this boosts tissue regeneration. Again, CoA-treated mice show a strong induction of olfactamine 4 positive stem cell at the bottom of the intestinal crypt after injury. So CoA is clearly an important downstream mediator of cysteine, but where is the CoA coming from? Is it coming from the CD8 positive T cells? Is it coming from the epithelium or from some other cell type? So to address this question, Feng Tao made a conditional knockout of SLC7A11, which is the cysteine transporter. So what does he find? So first of all, in data that I'm not showing you, when he knocks out SLC7A11 and CD8 positive T cells and places these mice on a cysteine-rich diet, it has no effect. So the phenotype is intact. But what happens when he now knocks this transporter out in the epithelium? So loss of SLC7A11 clearly leads to loss of expression in the epithelium. In organoid assays, for example, where he's knocked out SLC-say 11 and places the mice on a cysteine-rich diet, he now blocks the regenerative effects in culture of the cysteine-rich diet. And again, when he looks at CD8 alpha beta T cells, when he knocks out SLC-say 11 in the epithelium, he blocks those effects. He blocks the production of IL-22. And again, most importantly, in our regeneration assay post-injury, when he knocks this out specifically in the epithelium, he now nullifies the effects of the cysteine-rich diet. And this is important because this establishes metabolic coupling between the stem and progenitor cells and mucosal CD8 alpha beta T cells, where we propose the following. We propose that cysteine through SLC-say 11 increases the CoA pool in the stem and progenitor cells of the crypt. This CoA activates CD8 alpha beta T cells, enabling them to engage in IL-22 program. And it's been shown that IL-22 in times of injury is a very potent trophic factor that drives stem cell mediated repair. So we think that this is a novel paradigm in which small intestinal stem cells are coupled to a particular amino acid for their function. So with that, I'd just like to thank our many collaborators. I acknowledge the lab members whose work I presented today early on in my talk, and I just like to thank our other collaborators in Boston and throughout the United States and the world. I'd be happy to take some questions.